Hello, my name is Elena Swartz. It is October 30th, 2025, and I am presenting our application activity for Module 3 about how prokaryotic cells and eukaryotic cells interact with one another. We learned that bacteria, some common areas that bacteria are always present on the body are like our gut microbiome, our immune system, our skin microbiome. I chose to research how bacteria on the skin interacts with eukaryotic cells, specifically Staphylococcus aureus. S. aureus is a pathogen that commonly inhabits the skin. It can lead to bigger issues if it causes an infection while interacting with eukaryotic cells. An infection of S. aureus on non-lesion skin may lead to topical infections like atopical dermatitis, which is what I will be talking mostly about today, or it can lead to greater issues if infected on lesions, so get deeper into our skin and our body and our blood. So S. aureus can be in a colonization state, so that would be harmless. It's just part of the skin microbiome. A way that it turns harmful is when a biofilm forms. So a biofilm is the assembly of microbes attached to the target surface and then enclosed in the extracellular matrix. The formation of a biofilm triggers virulence factors like toxins or enzymes. Specifically with S. aureus, it's proteases and superantigens that really cause a lot of the damage. And then cause dysfunction of the skin barrier or soft tissues around it. So if S. aureus goes beyond the surface of the skin due to lesion, it can lead to bacteremia and sepsis of the soft tissues around it. So some of the virulence factors that occur in atopic dermatitis, like I mentioned, release of superantigens, which these trigger basophils and mast cells that release histamine. Many of the virulence factors trigger an immunological response deep below the skin surface, which is what I will mostly be talking about. So here is my first kind of picture. This is a picture of our Staphylococcus aureus. It's a spherical bacteria. It's gram-positive, so I drew in a thick peptidoglycan cell wall right here. I also want to make note that I didn't put any fimbriae on it, as the Staphylococcus aureus does not have fimbriae. But they do have that capsule or slime layer to help adhere to surfaces, and then also it helps form that biofilm as it's taking in information from the cell it's adhered to. I also drew a skin cell right here. So a skin cell is our eukaryotic cell. It has all the primary organelles, our nucleus that holds the DNA, our mitochondria for cellular respiration, our smooth and rough endoplasmic reticulum, and then ribosomes for protein synthesis. So we'll see how these two interact in the next picture that I have. Okay, so here we have our stratified squamous epithelial cells. So this kind of shows our skin, and then very zoomed in would be our skin cells. So way over here we have our Staphylococcus aureus. This is not harmful. It's kind of living on our cells, interacting with our cells here, but nothing quite serious yet. And then here we see that a Staphylococcus aureus cell adhered to the surface of our skin cell, and then it started to rapidly produce and form a biofilm with that extracellular matrix. So when this occurs, superantigens are released from the Staphylococcus aureus, which can cause a lot of inflammation. It can cause the Staphylococcus aureus to get into our skin. And so then it releases basophils and mast cells, which then increase histamine and as well as IgE, and then that causes those symptoms we see of redness and irritation. So here we can see how the cell and the bacteria is interacting with one another. The Staphylococcus aureus is on top of our skin cell there with our the cytokines, and toxins are released into our skin cell. Neutrophils are also very common to help fight off that Staphylococcus aureus. Thank you.